all this marine snow, some people call it because it looks like snow, that are particles, but there's many more types of particles in the ocean. And I represent them with this drawing that I stole from Laura Alonso Saez, who's a colleague of us. And I will talk about the marine particle microbiome. And this is very relevant because, you probably know what happens with CO2 in the ocean. You know the phytoplankton fixes CO2, and this phytoplankton may be eaten by zooplankton, may die, may be lysed by viruses. Oh, okay, viruses are here. So, and there is like particles that are being generated because of the action of the food web on the phytoplankton, and this turns into particles. And then the microbes come, or the bacteria and the archaea come. If the bacteria and archaea decompose this particular organic carbon in the surface of the ocean, the CO2 goes back to the atmosphere relatively contemporary with the primary production. However, if the particles sink into the deep ocean, then it takes hundreds or even thousands of years to decompose. So this CO2 that has been fixed by phytoplankton is actually removed from the contemporary carbon balance of the atmosphere and of the planet Earth. So these particles are actually colonized by bacteria, and this is what we will do here today. Okay, if you are an oceanographer, you will tell me, well, but that's not the only way by which carbon in the surface ocean goes down to the deep ocean. I ask you to read this paper if you want to know many other ways. Like this is what I told you is the biological gravitational pump. There is other pumps, the eddy subduction pump, the mixed layer pump, the mesopelagic migrant pump, and the seasonal lipid pump. But when you end up calculating how much of the export rate in the ocean, carbon that goes down to the deep ocean, is responsibility of this process is much larger than the other processes. So this one is relevant, very relevant. Okay, now I would like you to imagine, take this, what was the name of the film with a submarine that you could put into your blood and it was small and you could see the cells of your body. Well, do that with the ocean and go and imagine how the microbial plankton would look like, and you would think that it looks like that. This is what we commonly see in the microscope, and we tend to think that microbes are like that, distributed in the volume of the water. It would be something like that. So these are the microbes. There's two here that seems to be coupling, and some others that are daughter and son. So there might be some that are together, but most of them, they are isolated. However, the structure of the plankton is not that one. The structure of the plankton is something like that. This is the famous painting of Farouk Azam that has been on and on for years, saying that maybe the plankton looks like a ball of organic matter with lots of bacteria on top, dying phytoplankton with lots of bacteria on top, and so on and so on and so on. So in fact, you can actually go and look at the microbes under the microscope, and they are on particles. They are growing on TEP, which is something that is not really a particle, but it's it's a substrate that is difficult to stain. You have to stain it with specific stains. Sometimes they are living close together, as some of you know very well, because you have worked with the transfer of matter and energy between them, and so on and so on. And this is a new version of that same graphic that Roman Stocker's lab put together, saying how probably it looks like the plankton. So the plankton looks like that. So they are actually in groups. And not only in groups, but they are in groups where the food is. Well, I would say the drink is. The beer and so on are in this place. So this is how we should think about the plankton in that way. Another story that is worth to say is that the plankton, I mean, there is always this distinction between what is particulate and what is dissolved. And this distinction is operational. So somebody said, if you filter by this type of filter, what it goes through is dissolved, but it's not really the case. It's simply a continuum of sizes of particles, but we simply decided that DOC, dissolved, was from that size, and POC was from that size. Anyway, most microbes happen to be just across that line, okay, in terms of size. And when you put them together, I mean, there is, okay, free-living bacteria, which I will paint in blue from now on with the happy prokaryotes of Clara Ruiz. Thanks for the happy prokaryotes. Or they are attached to particles in which I will paint them by red. And they can be associated to zooplankton, to phytoplankton, to macrogels and transparent exopolymer polysaccharides, attached to fecal pellets, attached to dead organisms or fragments, houses or shells, other mineral particles, sand, clay, empty diatom frustals, and so on, so on, so on. So this is a division in which we separate those that are attached to particles from those that are free-living. And I will use this word very often. So what I will do, unless I talk too much and I happen to run out of time, is talk about five different aspects of the research we have done in the last years, and the research that we are doing right now and will do in the near future. How to study the prokaryotes on particles? Okay, if you are very rich and very smart, you can do something like that, which is a particle, an artificial particle that gets colonized by bacteria. The bacteria grow on the particle, decompose the particle, and by the time the particle is decomposed, I mean, the cells that were colonizing the particles are spread all over the place. Well, we cannot use those methods yet, but that's very nice, so I put it here. So because we have artists in the talk, so I wanted to. Anyway, so how we can study that? Well, very often what we do is what we call serial filtration. So we use different types of filters, and we separate what has a large size, and we say this is bacteria or prokaryotes associated to particles, and what it goes through the last filter, this is free living. But there are other ways of doing that. This is the marine snow catcher, which we just bought one and last week arrived, so it's here in the institute. And we will test it next week, so maybe we lose it and it doesn't come back. But anyway, this is a marine snow catcher. It's just a big bottle that after we take the sample, we let it sink, sediment, in the ship, and the particles go down. And you can either sample them here, or at the very bottom, if the particles are larger or not. Then we have the in situ pumps, and we have one here in the lab too that we just bought, thanks to Severo Cho. And this is something that you deploy to 4,000 meters if you want, and it filters in situ. So you get back with the sample ready. You don't have to refilter. And the other way of doing that is sediment traps. So those are things that collect everything that's falling down from the surface. We don't like them very much, and I will tell you about that, but later. So how many size classes? Well, Mireia Mestre, in her thesis, she put together some data that was, well, everything that she could find in the literature. And very often the people separate free living from particle attached just by using 2 filters at the most. So the 0.2 that collects the free living, and then 1 micron or 3 microns or 5 microns to collect the particle attached. Very few people use 3 filters, and very few people 4 filters. And the number of studies that use the filters, as you can see, everyone uses the filter that they like the most. I tend to think that everyone uses the filters that they find in the laboratory, but anyway. But there's kind of a tendency to use the 0.8 and 3 and 5 microns. By the end of the thesis, there were already a couple of papers that had used 3 or 4 filters, so that was a bit more sophisticated. And do all methods measure the same? Well, no way. That's very nice, very beautiful. Vienna compared bottles and in situ pump, and this is a distribution of several samples, and the bottles are the closed symbols, the in situ pumps are the other ones. This is a multidimensional classification of a community, and you can see that the in situ pumps are different from the bottles, but it depends on whether this is a small fraction or a large fraction. So in the case of the large fraction, they don't look alike at all. So in fact, for the large fraction, I moved it from Keynote to PowerPoint, and PowerPoint is so bad that it doesn't really take the images well. Anyway, so, and you could see that depending on the amount of carbon that there were in the system, there was a difference in the similitude between the in situ pumps and the bottles in the small fraction. And when you look at the composition of the community, just look at these two here. you can see that using an in situ pump or a bottle is like completely different. So very nice. That's why we bought in situ pumps. Anyway, so what are the patterns of prokaryotic diversity along the continuum of particulate matter? Are free-living and particle-attached communities similar? So they attach, again, the red, the blue, the free-living. So here, I would like to first introduce the system that Mireia designed, in which it was a sequential filtration. So water would be filtered by a 200,000 micron filter, then a 20 micron filter, then a 10 micron, then a 5 micron, then 3 micron, and so on and so on and so on. And those are some images of what happened in that sample. And just for you to know that most of the bacteria are in the last fraction. About 8% in Blanes, this is the average of the whole year, go above 0.8 microns, only 0.2 above the 3 micron, only 0.2 also above 5 microns, and so on. So most of the bacteria are here, and quite some are here. And now the question, which most of you know, so I'm not going to do what I do when I talk to people that don't know, is what do you think? The diversity will be higher, or bacteria will be higher in the small size fraction or in the high size fraction? Think about that. Those of you who know the answer, don't say anything. Think about that. I'm not asking to raise your hands. That's how it looks like. So the larger is the fraction, the more diversity, the more different types of organisms we have. In fact, this is the funny thing that happened. In fact, when you look at the structure of the diversity, you can see that the larger size fraction has many more rare bacteria. This is a rank list of the, I mean, you put the first one, the most dominant one, the second one, the third one, so on, so on, so on, so on. And however, when you look at the 0.2 micron, you see that there is more dominance. So there's a few that are very abundant and quite a number, quite less rare organisms there. And when you look at the groups, and this is 4 classifications that had to do with the different filters. Red is more abundant, yellow is less abundant. We could differentiate organisms that were always in the free-living size fraction, others that were always in the second category, so in the 0.8 filter, others that were all over the place, like these ones, or some other groups that were always in the large size fractions. Some very obvious, like where are my beloved Beromicrobia? Well, they're not here. But Beromicrobia were one of the ones associated to particles. Okay, does this change with time, and does this change with time? Well, what we found was that this structure, this is a whole year, all the fractions, different colors of the fractions here, different colors of the months. You can see that there is a seasonal divide. Blue colors, winter, red and yellow colors, summer, it's very differentiated. But this differentiation based on the size part of the particles changes continuously too. And when you look at which factors define the structure of the community at each of these size classes, you can see for the small size classes is something like phosphorus. Well, temperature everywhere, chlorophyll everywhere, day length everywhere, but here is phosphorus. And when you get to some other size classes is depth, the depth of the Secchi, which is a measure of the turbulence, the transparency of the system, the number of particles in the turbidity, that's a word I was looking for. And when you go to larger size classes, it's also salinity that plays a role. So there's the different factors affecting. Then we went, well, in fact, it was before, but for the story is then. We went to the Malaspina, we took the ship, we went all around the world, and well, there's tropical and subtropical oceans, and we collected lots of samples. We were filtering lots of water, different types of filters, so on, so on. I'm not going to explain. And we were interested in the deep ocean. And the reason we were so interested in the deep ocean is because the deep ocean has a very important biogeochemical role. I mean, production and consumption that's happening there is very relevant for the whole of the ocean biogeochemistry, because there was evidence for a dominant particle-attached way of life in organisms that had been isolated and that were demonstrated to live very well under high pressures. And because there was lots of dissolved extracellular enzymes, And if you're a bacterium and you throw your enzymes outside, the enzymes that you use to collect carbon, I mean, that doesn't make sense if you live alone, right? So it makes sense only if you are living tightly packed with your friends and your daughters and your sisters and all your family. Then you can put in common all the enzymes to develop. So there was quite an evidence that the particles were relevant. This is what we found in Blanes Bay. However, we went, a PhD student from Italy that spent some time here, went to the northern Adriatic Sea, did the same. She did not find the same, as you can see. Free living had more diversity than particle attach. Then also Mireia went to a cruise and did the same in the surface, in the DCM, in the mesopelagic, in the bathypelagic. Those are the size classes. And you can see that this is not so obvious that the larger size classes have the most diversity, so it tends to bend and have a maximum of at some sizes. Those were oceanic stations, so they were different from Blanes. And now the question is what happens in the deep ocean all over the world at 4,000 meters. We could do a poll, but we will not do it. I go straight. Here we found that the richness was higher in the free living fraction, and the phylogenetic diversity was also higher, but the phylogenetic diversity per unit organisms was actually higher in the particle attach. So the particle attach organisms there were less of them, less types of them, but each one of them was more different than the others. That's what this figure shows here. And just to give you a glimpse of which organisms live in the deep ocean in the free living fraction, well, this is an archaea, this is a delta proteobacteria, this is Alteromonas, a gamma proteobacteria, and so on and so on. And those are the ones, oh sorry, this is the free living, yes. And this is red, so this is the red. And the dominant one is an Alteromonas. Wow, wait, Alteromonas. This will come back later. Then actinobacteria, then other gamma proteobacteria, and so on and so on. And this is very interesting. This is all the stations of all the oceans. This is the different oceanic basins. Again, in blue, free living, in red, particle attach. The first most abundant O2, Alteromonadacea, as you can see, was about the same everywhere in the two particles, except here, for some strange reason. However, the second most abundant was only in the free living fraction. The third most abundant was only the particle attached fraction, but in some of the oceans, not in the others. And the fourth one was exactly the same, in exactly the same abundances, but in some oceans, and in others, they didn't appear. So this is interesting. One of the things that Guillem, who's the student that did all that, realized, I mean, he plotted a phylogenetic tree of all the bacteria that we were seeing, which is this thing here. And then he plotted 3 lines. The blue line was if this organism was developing in the free living fraction, and the red one is whether it was living in the particle attached fraction. And he noticed that there were whole phylogenetic groups, like that one here, that were only or dominantly in the particle attached fraction, and whole phylogenetic groups that were only in the free living fraction. So he wondered what makes an organism be, I mean, to be particle attached in the deep ocean or free living is something that is in your phylogenesis. I mean, it's because you are this guy and this guy always is in that fraction or not. So he did this very complicated thing, and he computed organism by organism the phylogenetic distance, and at the same time he computed organisms by organism the distance in their lifestyle. So he gave 1 to the organisms, individual organisms that were in the particle attach, and 0 if they were never. So, and you can see that there is a correlation between phylogenetic distance and lifestyle distance. And then he plotted this line, where it breaks the correlation, he plotted that. So organisms of the same genus were all correlated between there was always a correlation between lifestyle and phylogeny. In the same family, the same. In the same order, the same. In the same class, basically the same, a few exceptions. And then when you got to phylum, then it broke down. So if you're in the deep ocean, if you belong to a given class order, we know whether you are particle attached or free living. Then what he did, he constructed a distribution of the ocean basins, which I only put this graph because you remember this figure. And then he plotted the different groups on top of this figure. And you can see, for example, Deferribacterias were all free living, Firmicutes were all particle attached, Actinobacteria, oh well, they were in both places. Microbia, my favorite ones, were mainly in the free living, and Euryarchaeota were all in the free living fraction. So you can plot the different groups and select those that are in the free living, like in blue, those that are always in the particle attached, like in red, and those that are who knows what, in green. But just remember that to be in green, like a whole group like Delta Proteobacteria are in green, might mean that there are two different groups, like OM27 and SAR324, that have different patterns of habitat or something like that. I'm not going to talk a lot about that, but we also did metagenomes of these samples. And this is a comparison of the number of stations that were collected by the Tara Oceans with depth, and those are the ones that we collected in Malaspina. And we did the metagenome, so we looked at the genes in these different stations, and just to give you that there were about 40% of genes that were all Malaspina only in the free living fraction, and a very variable number from 20 to 80% of genes in the particle attached fraction that were only appeared in the databases of Malaspina. This is changing continuously. This is what, the day it was done. And this is, I didn't know how to show that visually, so you know that thing? So when you went to the Department of Biochemistry to do the practical work in the Department of Biochemistry, they had all these posters that you thought, well, they could have, I don't know, soccer landscapes, and they had this thing. Well, they love it. So this is all the biogeochemical pathways, and in red are the ones that we could detect in these samples. And I'm going to move between the free living, this is the free living, this is the particle attached, the free living, particle attached, free living, particle attached. And you can have a glimpse of the differences in genetic capabilities of each one of these fractions, okay? Okay, and not all genes were important in all fractions, as you can see. This is the same type of graph. And I'm not going to go further into that because that's a... Oh yeah, that graph. Marta Sebastian, in one of the papers that is under review, showed that there were some genes, or some groups of genes that were more common among particle-attached organisms, such, for example, carbohydrate metabolism, for example, elemental information processing. This means interactions between organisms. This goes here. And this one, which was glycan, is not very relevant. And others, like, for example, vitamins, or genetic information processing, or energy metabolism, that means unusual forms of energy capture that were more common in the free living fraction. But I'm not going to go further into that. So following with the work that Marta has been doing in the last years, we also were very interested in how it's like the life of prokaryotes in the deep ocean. And, okay, so this is the different places where, the different types of food that the bacteria in the deep ocean may have. Particulate organic carbon, particles, dissolved organic carbon, suspended POC. This is a bit difficult to say. This is particulate carbon, but that doesn't go into the traps for some reason or another. And then labile carbon that's being created in situ in the deep ocean. So the POC is very sporadic in time, and it's very difficult to predict when there will be an entrance of carbon. This is just the POC flux into the deep ocean in one given station, and you can, through the year, and you can say that there's some peaks once in a while, but there's whole seasons in which there is no inputs of particles. They suspended POC, nobody knows very much what they are. Some people say that they have been produced in situ. Some people say that this is carbon that comes with lateral advection, and it gets into the samples, but it comes from like areas with upwelling or things like that. And the labile carbon would be carbon that is being produced by chemolithoautotrophic metabolisms. That means primary producers that do not use light, but they use chemical reactions to generate the energy needed for growth. That would be the different methods. So by the time she produced this paper, people claim that most bathypelagic heterotrophic metabolic activity is mediated by particle-associated prokaryotes. Look at this paper that says, yeah, predominantly particle-associated lifestyle of these C prokaryotes and so on. But we have seen that free-living and particle-associated communities are distinct. I've shown you also that the lifestyle has been conserved throughout evolution. And in a very nice experiment, Marta demonstrated that deep ocean communities can handle long-term starvation. So for these experiments, what we used was a big tank, one of these tanks for people keep water for gardening and things like that. And after cleaning it very well, we filled up with bathypelagic water, and we decided to run it for a year. And here is a whole year. So she was very constant and took samples every once in a while. And you can see that DOC didn't change here. There was not much. There weren't particles because it was closed, so there were no more particles coming in. It was closed. DOC was constant. And what we observed was that what we expected was that activity should go down like that, abundance should go down by that, and diversity would most likely go down like that because there is no input of energy into this system. However, the reality was that the total activity went down, but the abundance did not. After about 3 months, the number of cells increased in the tank, and at the end of the year, we had the same number we had at the beginning. And the same did diversity. So what happened was that between in this change that we have seen here, sorry, what is that? This change here, this point here, there was an increase of archaea, and this archaea most likely, and the community before that peak and the community after the peak was very different, as you can see from the colors. And we came up with a hypothesis that what happened here was that there was some carbon in the community. This was spent by the organisms, and then it was like slowly nothing happening until there was a moment that Taumarchiota started to develop and created organic carbon using chemolithoautotrophy. And out of the carbon created by the archaea, then other organisms could develop in the tank. So the tank has life. The tank actually evolved in the way that probably resembles what happens in the ocean when there are no inputs from the surface. So this involves the metabolism, this involves the metabolism of nitrogen and nitrite oxidizers, the archaea, and the use and the production of the dissolved organic nitrogen, thymidine, vitamins, and so on, that fed the heterotrophic prokaryotes. This had been discovered, that this existed in the deep water, but that sustained the community and make the community change, evolve in the deep ocean. This was something new. So then Marta went to test the hypothesis that activity is associated only to particles. And what he wanted to do is to assess the contribution, the heterotrophic activity, of different lifestyle categories to the active microbiome. And the idea was that particle-attached microbes live out of very labile carbon, so fresh particles come, and particle-attached microbes use them. And free-living prokaryotes live out of recalcitrant leftover carbon that is there. However, as I showed you before, we observed that there were groups that we could see them both in the particle-attached and in the free-living category. So called differential abundance analysis, in which she could divide the organisms depending on whether they were always particle-attached, always free-living, or sometimes particle attached, sometimes free living. You can see the numbers, 500 versus 2,000 versus 900. And this is the distribution through the stations of Malaspina. And you can see that the dual ones are about, well, 30% or something like that everywhere. But in the active fraction, because this is the total organisms, this is the number of active organisms, they were a bit more, they were about 50%. And the blue ones, which were the free living ones, were only 20% of the active organisms. She also found, and also in Malaspina, that this is the particle attached, this is the free living, that the relative abundance in the active bacteria was changing depending on whether they were living in stations dominated by labile carbon or stations dominated by recalcitrant carbon. And you can see that depending on whether the carbon in the deep is more fresh, more labile, you could see more of these organisms, and more of these organisms free living where it's more recalcitrant. So it looks like she put it together. Well, no, I'm not going to jump this one. This is fresh. I stole that slide from her yesterday, but I'm not going to talk about that. So she will talk about that some other day. She defined that there were probably particle-attached prokaryotes, that they were fast growers capable of uplifting up the metabolism, so responding fast to the changes in the community, and they develop in waters enriched in labile carbon particles. Free-living prokaryotes, they were slow growers generally. They had limited capacity to respond to changes, and they thrive in waters depleted on labile organic carbon. And then there was the dual lifestyle prokaryotes that were like the cars that the hybrid cars that can use electricity and petrol, that could thrive also in waters with labile DOC. And one of the things, the ideas that I put forward, that I was insisting, but we had no evidence to demonstrate, there had been many years ago a paper by Farouk Azam talking about the baby machine. And he said that the particles were baby machines. The particles were producing babies, throwing the babies outside, and letting the babies colonize other particles and develop in the other particles, which would be something like the video that you showed before. But we didn't have any evidence, so we didn't write it in the paper. So, and the final part is the relevance of particle colonization for ocean linkages and sinking fluxes. I'm finishing already. As I told you, the particles sink down out of the photic zone. So it could be that a particle that goes very fast to the bottom is very similar at 4,000 meters than it is at the surface. However, a particle that takes years in going down, it gets slowly decomposed, and it changes the structure of the particle as it goes down to the deep ocean. So this was tested because as vectors of diversity, vectors of microns to the deep ocean by Mireia Mestre. And what she did in several stations in the Malaspina expedition, she looked at the surface, DCM, mesopelagic, and bathypelagic at different size fractions and compared all this. The interesting thing here is that she started labeling with yellow color those organisms. I mean, she assumed that it went from the top to the bottom, and labeled those organisms that appear at the surface in yellow color, those that appear at the DCM in green, those that appear in the mesopelagic in blue, and those that appeared that were new in the bathypelagic in dark blue. What you can see here is that most of the deep ocean organisms were already there in the surface. So what it's saying is that likely particles get colonized in the surface ocean, and they go down as they are. They change a little bit when they go through the bathypelagic, mesopelagic too, but at the end, most of the bacteria in the particles in the deep are actually from the surface. And they are not always the same. So if you look at these seeds, so these ones that colonize the particles, they are, the only thing I want you to look is the colors, and you can see that in different stations, it's different type of organisms that are developing there. I'm jumping this one. Well, no, I'm using this one. Then Clara, who had been very involved in the previous study, she's a strong-minded woman, and she didn't let that thing go. She wanted to go further into that, and she looked at the stations, and then one day she comes and she says, wow, look at that. The large size fractions are very well correlated to things of the surface, more than to things that happen at depth. So what happens in the surface, like productivity, chlorophyll, different types of organisms, and so on, is very much correlated to the communities in the bathypelagic, and what happens in situ, well, not that much. And she went further and presented this graph, which is very complicated, so I'm going to explain it, but then I'm going to simplify it. So she put primary production, which was taken from atlases, and then chlorophyll concentration, which was again taken from atlases, and calculated the vertical dissimilarity. Watch out for this, vertical dissimilarity. So more dissimilar at the top, more similar at the bottom. I know, I don't know why they call it like that. I mean, this is making things very complicated all the time. And she did that for the different fractions, the small ones in blue and the large ones in colors. You can see she put the lines everywhere, but the asterisks tell you only those that were significant. Let's simplify. In the large particles, there is more similarity in the particle-attached communities when the chlorophyll in the community is large. And there's less when the community is low. The same happens. And then that's a graph that Vienna, which Correa put together, came the hypothesis. So if we are able, so if we have stations that have a high carbon flux, so lots of particles and they go very fast to the bottom, then the communities will be very similar in the top and in the bottom. However, if the flux is very slow, then the communities will be very distinct. We can turn it around. Measuring carbon flux is not simple. However, measuring the structure of the bacterial community in the surface and at depth in particles is relatively much easier. So let's put the hypothesis that we can measure the vertical dissimilarity between the particle-attached communities to estimate carbon flux. And this is the first proof of concept that this may happen. Vienna went to these two polynyas in Antarctica and measured exactly that. Unfortunately, she was alone, or somebody that helped her a little time, and the crews didn't move from the same station, so she could only do that in 3 stations. The 3 stations were different. This is the communities in the 3 stations, the free living, the small particles, and the large particles. You can see that there are changes. The 3 stations had different chlorophyll, different POC, different flux of thorium, and I'm going to explain that in a moment. And number of surface O2s in the different stations, which is the same order than here, were very different. So in the particle-attached fractions, there were like 60% of the O2s, or organisms, at depth that were already in the surface. And in the medium station, well, about 30%, and in the other station that didn't have that much flux, it was very low value. So that was the hypothesis, those were the results. So in fact, as we increase, this is an estimate of carbon flux from thorium, we'll talk about that later, or you can ask Vienna more, that she knows better. Those are the estimates of carbon export flux, and this is the vertical dissimilarity of particle-attached communities. So when the flux is very low, the communities are very alike. This is vertical dissimilarity, maximal dissimilarity. They're very distinct. When the communities are, when there's a lot of carbon export flux, there is much more similitude between the communities. And we tried to put that into 2 projects. In one part Vienna put that into her junior leader, and we put the hypothesis as one of the hypotheses of Bicolor, the project that we have now, which came from an ERC failed project called Procapart, which I submitted, and it went a little bit farther, but not a lot, but also with a project that Laura Alonso, former PhD student of the ICM, that it's now in Austin in the Basque Country, that she produced a consolidator grant that was not funded, it was called CellArt. And then we involved Olga Sanchez from the Autonomous University, and we already have Paula Sabaté as FPU to work on the project. And what we are saying here is, look, does it matter if we have a particle that's colonized by the blue guys or a particle that's colonized by the red guys? Will they sink differently? Will they produce more carbon export? Or it will be the same? Does it matter whether we have lots of isolates of those that Ramon likes so much, or we have yeast developing fungi, developing in the particle? Does it matter? Then the part that we are not so much involved, but Laura Alonso is very much involved, is how do cells communicate in the particle, which tools they use for quorum sensing, for elaboration of EPS that bridges or brings together the particle and makes it more solid, that will sediment faster, or some enzymes will facilitate that the particle is loose and doesn't sediment that fast. And which enzymes do they play in a role here? So that's part of the work that mainly Laura will do. And then this is work that Massimo Pernici did for her thesis with Ramon, and those are the distribution of eukaryotes on particles in the Malaspina. And yes, there are some places where there were lots of chrysophytes, some places where there were lots of Spumellarida, which, if I'm not wrong, they all feed on bacteria. Correct? Basically correct. For this talk, it's correct, right? Okay. Thanks, Ramon. However, look at these stations here. Basidiomycota. If you ever went to pick up mushrooms, you know perfectly well what are the Basidiomycetes, because they're the ones that you feed on. Right? So we had fungi here. Not that we had rubellons down there, right? That has nothing to do. Anyway, so this may play a role. Also look at this one. These two bacteria were isolated by Laura from particles, ocean particles. They are very similar phylogenetically, Pseudothermomonas, Alteromonas. But look how different they behave. This organism does not produce an exopolysaccharide matrix. However, it produces lots of vesicles. This one doesn't produce vesicles and produces a hell of a lot of an exopolysaccharide basin. So, and then immediately when I saw the image, I thought about the teeth and the people that work with the bacteria that grow in our teeth. These bacteria develop a microfilm on our teeth, and depending on whether you have this microfilm, you get your teeth decomposed. And then you have to pay a huge fee to the dentist. Right? So then we put together the hypothesis that depending on which organisms and which production of this exopolysaccharide, it will have a more compact or a more diffuse or more less compact particle, and this make a role. Meaning that this idea here, that maybe, yes, it does matter whether it's the Pseudomonas or it's the Pseudothermomonas, the one that is colonizing the thing. Then we will do other things. We'll collect particles from here. We'll put them on dialysis bags, and we'll incubate the dialysis bags in our famous bathypelagic or mesopelagic or whatever dark ocean tanks. This we will not start yet because we're waiting for the FPI person, but that's the job that she will do probably. And the other thing that we will do in this project is that we will separate the particles with different ways. That's the Mireas setup. Those are the in situ pumps, and those are the marine snow catchers, which separate between slow sinking and fast sinking particles. We will do metabar coding, interaction networks, metagenomics, isolation of prokaryotes, and so on and so on and so on. But we will do that because there's lots of people doing that, but not that many people that simultaneously measure the biological carbon pump, and that's the key of the project. If it succeeds, that will be the key to success. If it doesn't succeed, we will find another excuse. Anyway, the idea here is that when you measure particle flux, you can do it with sediment traps, but this tends to give you the values for days. optical measurements, and we are working with Marty and thinking ways of implementing methods, but this is not that clear yet. Or the thorium-uranium flux method that measures the flux over weeks and months. The idea here is that you measure the thorium and the uranium in the water column in the particles, and what happens is that they all come from uranium, which the uranium decomposes into thorium, but the thorium is scavenged by particles. So you can actually measure both and plot the 1-to-1 ratio, and this area here is the deficit in thorium, and it's directly relatable to the particle flux, the carbon flux. So the idea here is that we measure both things simultaneously, which is, I think, what would be the key. So I'm finishing a few take-home messages. People use mostly with just one filter to separate free-living from particle attach, commonly these sizes. In the bathypelagic ocean, free-living communities are more diverse than particle attach. It is not the same in the coastal ocean, and it varies in the open ocean. The genomes of particle attach and free-living communities are very distinct. The different lifestyles appear to be quite conserved phylogenetically, at least at the class level, using various size fractions and Bell's patterns of diversity with size and groups associated to each size. Deep ocean particles are colonized at the surface. The more, the faster stuff sinks, the more similar are the particle-attached communities of surface and bathypelagic ocean, to be further tested, and working on coupling estimates of carbon flux and particle microbiome characteristics and what's going on in them. And this is what we are doing next week in the groups that we have. If you want to read more, I try to give an overview of all papers in one talk. I don't know if I succeed, you will tell me. And thanks to all the people that run Blanes, that run Malaspina, some people on the bioinformatics side, and all the projects that pay different parts of this story. And that's all for now. Thank you.
Okay, Pep, many thanks for this talk. Now we have some time for questions.
Thanks, Pep. As always, a very inspiring, exciting, and interesting talk, and long, and many, many, many slides that you manage very well. So my question is about the experiment of this big tongue, where you only talk about bacteria, but I am wondering if you saw some predators as protists or viruses that could maintain these different prokaryotes in different abundance and different diversity.
If I'm not wrong, but Marta might correct me, we did not look at the viruses, but we did look sometimes at the eukaryotes, and we didn't see many things going on.
Well, another hypothesis...
There were other tanks that are running now that had eukaryotes actually using organic carbon, but that's another story. Yeah.
But I don't know, but another hypothesis could be that some of these viruses could select for different type of bacteria, and then just the ones that were not infected are the ones that they cover.
Yeah. Does it make sense for a virus to, in a community that doesn't have carbon and it's not growing, has very low activity, to kill your host and go out into the wild?
Well, why not? This is the piggyback the winner, I mean.
This is the suicide strategy for a virus, right?
Well,
so
I don't think so.
It doesn't really make... However, the decline of the archaea may have been produced by viruses. I agree with that. So there was a bloom of archaea, and maybe the archaea simply disappeared.
But then this will promote more organic matter that will be able to take for another.
That's why the community changed after the increase in archaea. So I think that up until the increase in archaea, this was probably just the bacteria that decided, or the prokaryotes that decided to do what they wanted. When the archaea were developing, it's very likely that they were killed by viruses. I wouldn't, or maybe the low number of predators that were around. Yeah.
And another comment in this wonderful project that you have, that you will consider fungi in these attached particles that you have bacteria, eukaryotes. Why you don't consider viruses? Because if you have if you have a film of bacteria there attached, if one is infected by the virus, it will multiply exponentially.
Well, this is a good discussion that we can have, but you know these lakes that have blooms, very concentrated blooms of cyanobacteria or photosynthetic bacteria, Chromatiaceae. They have a bloom, a huge amount, and it stays there the whole summer. So viruses, for some reason, they don't want to kill a winner. And the reason why the viruses don't kill that winner might be because they are already all there integrated and waiting for the bloom, the fall overturn, when the lake destabilizes, things like that. So, first thing, I'm not sure of what you say that the viruses may be very relevant, but I do think that we have to look into them. But we will have very nice metagenomes that you will be able to use your fantastic
in the future we'll have.
And methods to inspect. In fact, you will
with the collaboration of
the
Felipe.
next cruise. I mean, he will come to the next cruise, so he will see it in reality. Yeah.
Okay, and rely in Felipe.
Well, I'm not going to ask about viruses for now. I was curious about, like, you have this size fractioning, and you show that the diversity of the communities change a lot. But I wanted to know about the absolute number of cells in each one of these fractions, and if that changes with depth or if it's more or less stable.
We don't know this. We don't have this data. There was a student that was supposed to collect this data, and that was her project. But we had lots of troubles doing it in Blanes. So I show you averages because that was the only thing we could get in Blanes. And in the Malaspina, it simply didn't work because, I mean, you count the total and it's 10, and then you count the side fractionation and it's in the different fractions and it adds up to 15. And then you say, I don't know what's going on here. So we need to have a better way of measuring or counting or enumerating the organisms on particles. So it was not done in Malaspina because of that reason, because it had not worked well in Blanes. But it's really, this is a failure that we have and that we should, nobody has come up with a method to do it.
But you counted through cytometry or microscopy or something? Yeah,
we did the fractionations and then counted the cells that were retained. So you took the maximum and then the retained. But if you have the maximum is 100, and then you have 80, you say, yeah, 20 were in here. And then you have 30, yes, and then you have 100. And then you have 200, that doesn't fit, right?
Thanks. There's too much information to digest. Even if you more or less know what you are going to talk about. But thanks for all this full of information that gives me a lot of questions. And one is a methodological one, which is when you say that there's different, the diversity of bacteria between the C2PAM and the Niskin bottles, you only have the data of diversity of bacteria, but I'm wondering if you, maybe it's done, if the comparison of the size distribution of the bacteria, of the particles, size distribution of the particles of the CT-POM and the Niskins. And my question is related to maybe they are broken, blah, 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 and then how they look, just the size distribution, no? To put in context the
it could be done with bottles, but for the in situ PAM, you don't know what it's filtering. I mean, you drop a pump there and open the pump and wait for 2 hours and it comes out. Yeah. And you assume that it has filtered everything that encountered. It could be perhaps correlated with some optical measurements. So a couple, an optical, you know, the underwater vision profiler and all these kind of things. That
Or resuspend the particles in the filter and
yeah, but you know that you're not going to resuspend the small ones. You know that. And the large ones maybe stuck together.
well,
So.
but this is what you are going to analyze.
Yeah, I know, but no, what I'm meaning is that if you resuspend, in fact, Vienna resuspends some of the large filters for some of the measurements she does. So she assumes that it works, but we have used this resuspension method sometimes and it didn't work very well. So yeah, something to think about. Yeah.
And more, not so methodological. You were talking always about the size of the particle, but what about the quality of the particle?
Yeah.
And
Yeah, that's the key.
because this difference between free attached that you have in different systems or different places,
I'm sure it comes from the differences in the quality of the particles. So, yeah, we are talking with Eva and with Maria Calleja to do some description of the carbon quality of the different particles, but it's far.
Hi. Pep?
Yes.
Coming back to the big carboy bathypelagic experiment.
Yeah.
Aren't you afraid about the plastic substances that can link that? For one side, it can kill some species, and for the other, it can provide food for others. So it can be the change of the species composition that you have there. It can be even explained by that.
The tanks. I mean, this is drinking water quality plastic. So supposedly people drink the water that is stored in these tanks. No, I'm telling you this story. I was once in Switzerland, and there was a research group that worked on the bacteria that grew on in the inner part of the plastic bottles of Evian. Mm-hmm. So the plastic bottles that Evian was one of the most expensive drinking water and so on. Well, they could cut the pieces of plastic. They were looking, and there were bacteria that when they were cut, that they would get inside like a worm inside the plastic. So.
Yeah, and if you use dialysis bags, they will fit on it for sure, 100%.
They will, sorry?
They will fit on the plastic of the dialysis bags because they love it.
Yeah, that would be a short experiment. So.
Okay.
We have used them in 3, 4, 5 days in eutrophic lakes, and they lasted. So we expected here to use them for a couple of weeks. But yeah, that might happen. Yeah, I agree with you.
So sorry I missed the former part of the talk. I was distracted by someone, so I arrived at I don't know if you've talked about this. So this works for open sea environments. We have talked about this several times. So you have lateral inputs as you get close to the continental margins. But that's my question goes more into the type of particles. So you have divided the large particle and small particles. That's very crude, a very crude division. But have you looked at the role of fecal pellets? So when I did my PhD, I started with analyzing sediment trap data and looking at the type of particles through the binocular. And most of the large particles, they're more dense and compacted. And in most of the cups, the majority of the particles were large particles were fecal pellets, and you can identify ellipsoidal ones and very like hairy type of and most of the flux was due to the at least the fast sinking due to the fecal pellets. Yeah. So and if you can comment on that.
If you find salps, it will be even more. In none of this work we looked at that carefully. But one of the reasons why we will do 18S sequencing on the particles will be to try to identify the origin of the particles. We will look at them too. And this time we are bringing a binocular scope and so on to the ship. We'll look at them, but we will also look at the 18S sequences to try to see. I mean, if if your particles were mainly fecal pellets, you will have quite a lot of 18s that corresponds to the zooplankton. So that would give you a glimpse of where the things are going. But yeah, you are completely right that if you have a station that's full of Copepos sheeting, you will have lots of these fecal pellets, and in another station maybe you have a diatom bloom, or maybe in another station you have nothing that you can identify. So that's also the reason why we also sample the rest of the, we also look at the surface, because we got to know what is going on at the surface, no? Not only look at the deep and see, no, we have to look at different depths and see if we can identify something. So here I only presented you, or only a small result of eukaryotes, but we will look at eukaryotes, maybe with 18S, maybe we have to use some of eDNA techniques for other organisms. We'll see how this goes. Yeah. Yes.
My question is kind of a follow-up. I also was interested in the nature of the particles and how that influenced the communities that might attach to it, and if you have data on if these particles are just like biological particles or also microplastics.
Yeah, we didn't look at, well, the first question is the origin of the particles. As I said, this was prokaryotocentric vision of the particles, so we didn't look at the rest of the things. Now we are going to look at the rest of the things. And particles, plastics, we didn't look at that, but we are convinced that there might be. And in fact, the plastisphere has been seen as also another mechanism of vector to the deep ocean. So there might be organisms in plastics, but we haven't looked carefully at that. So now we are trying to characterize the bulk of the particles with some chemical methods. It will take a while between I mean, well, Marta told me yesterday, we're going to do things we have never done, so this cruise may be a mess, and that's probably what's going to happen. So we are now going out 10 days to learn. And the next cruise, which is the large one, we hope we know better, and we can make choices and select different things that don't work and do it then differently. Yeah. But yeah. But not that for the empirical relationship between flux and particle dissimilarity, I mean, we don't care whether they are fecal pellets or they are plastics or they are whatever. So and in front of Celia would say, we don't care about the organic carbon and the quality of the carbon, we only care about the microbes, but that's just for teasing her, but yeah.
Well, thank you, Beb, for all the food for thought. I'm like Celia, I'm a bit overwhelmed by the breadth and depth of data and ideas that you gave. So, but thank you very much. I like very much the idea of linking the export flux to the bottom to the similarity or dissimilarity of the communities, and I think that would be an upwelling system, and going away from the upwelling would be a perfect way to test this, and that would be really nice. But I don't know, maybe you plan to do it as well, or maybe
the large cruise was supposed to be leaving in September, Vigo, so crossing the upwelling in Vigo, then going into the central Atlantic, and then back into the Canary Islands upwelling. Although this one is a bit messy because there's a lot of lateral flux there, it may be a bit of a mess. Yeah. That's the only thing we could put in the project without going nuts with the budget, those kind of things. Yeah.
But I wanted to comment on something that a picture that you put on a microscope picture with all the dots that were bacteria, and they were all there together, very happily and nicely together. And of course this is false, because you can filter 10 liters of water onto a small filter and they will be packed with bacteria. But the reality is in the ocean that the free-living bacteria are a distance apart. Like us would be like a kilometer or 10 kilometers apart. And it's very hard for them to find each other. It's very hard for predators to find them and for viruses to find them, just because of encounter probabilities. And there's where particles are very interesting and come into play, because there are these hotspots of activity and so forth. and it seems that all your work focuses on the bacteria, and as you answered Dolores with the viruses and so on, it doesn't seem that you give a role for predators and viruses to structure these communities, to infect these communities. While when the activities on particles, they could have a large effect, I think. But, and this gives some idea or philosophy out of this or whatever. It's like everything is structured bottom up in terms, substrates, bacteria, and then everything else is a subproduct of this structuring that comes afterwards. Is this what you think, or?
No, it reflects basically our limitations, our intellectual limitations, and our ways of thinking on experimental ways of addressing these issues, as you say. I think that there's this group in the States, Otto Cordero's lab, that does very nice work with model particles, and they follow the succession in the model particles. They are particles that they made of one substrate, and then they let them colonize by seawater bacteria and see how it evolves the community, and so on and so on and so on. I don't remember having seen anything about the eukaryotes or the viruses in those papers, but I didn't read them very carefully either. So I think that maybe the dialysis tank experiments could be a way of seeing what happens. The problem is that, can you follow a single particle? No. Can you follow a bunch of particles that are relatively similar? Maybe. But then you will not know what is going on in each particle. So we are also, in this time, we're selecting single particles and trying to do metagenomics of single particles. If this works, we could then think of ways of generating particles that are relatively similar and let them evolve individually and see whether proteins develop, and when proteins develop, there's a change in the community, and there's a change in the degradation rate and things like that. But that's, I would say, it's a bit far away. If you have a bright idea about the way of addressing that, we would love to hear, because we put some effort at thinking. When I was writing the ERC, every time I had an idea, there were my colleagues that came saying that, whoa, why, no, that's not going to work. So, you have to balance the things that can work with the things that would solve a problem. So it's yeah. I remind you that I have a sore leg. So.
Sí. I think probably there is. A last question for this very interesting debate. But Rafael, you ask me.
Hi, Pep. I'll be asking on behalf of Martí Galí, who's connected online, and apparently he couldn't access the chat. It's not activated or something. So Martí says, just a comment and question. I'm not surprised that filters and in situ pumps collect different types of bacterial communities because they likely collect different particles.
Yep.
Current estimates, he says, suggest that 80 to 90% of the POCs suspended and with sizes of a few microns, whereas fast-sinking particles are typically larger than 100 microns. And he goes further and says, and suspended particles, that the small ones, likely support most of the respiration in meso and bathypelagic waters. So the question is, are you planning, or is it any possible to use sequential filtration with in situ pumps to distinguish different functional fractions?
Yeah. The one we bought has three sizes of filters. The ones we will be lent by the Monaco team will only have two, unfortunately. But at least one of the pumps will have three. Yeah, that's the idea. The idea of the project is that using this method, we're collecting different types of particles. So we don't expect to look alike. We already test that and it doesn't work, so don't need to go farther. But the way in which we will collect the particles will actually be different types of particles, and then we will inspect whether one type or the other type of particles is more linked to the flux or things like that. So maybe what happens is that the amount of carbon and the community composition here compared to the surface is the one that is well correlated to flux. Or maybe it's the one that we collect here, or maybe it's the one that we collect in one of the filters. I don't know whether these are the filters or not. So, no, these things, I think. Well, anyway, whatever. So yeah, we'll do this job and see what happens. So this is very much Gasol's strategy always. So let's go measure most of the things, and then we will see which one is the one that we should have been measuring. But I cannot figure out another way of doing the things than that one, where we have no knowledge. Period.
And when you measure vertical velocity, because we are always talking about biology, I'm a biologist.
Yeah.
But to understand all this thing of surface and the communities are more different or less depending on how much surface is up there and things like that, I was thinking, is this because less particles are right there? And then, well, anyway, a lot of things about community and colonization of particles and sampling effort. And then I thought, okay, but it's very important the physics of the water column. Maybe you can start to distinguish.
Yeah, we assume that we will be sampling always stratified water columns in which the deep ocean is isolated from the surface ocean. So we may have a very fast.
Yeah, but what happens in the sampling?
Yeah. We might have in the epipelagic a very fast movement of water, and I don't think we could be measuring that with Bea Mourinho's thing, these profilers that we were using in Malaspina. I don't remember the name, but I'm not familiar with that, so I'm sure I will be able to.
Because like the time of sinking is related to physics, but also to the quality of the particles, and it's related to the community and that. So try to disentangle.
Yeah, but look at that. We don't try to disentangle all these things. We just go we are purely empiricists here.
I am an empiricist. What I say is that to try to relate the community structure effect on the quality of the particles, the physics of the particles, and then the sinking, I guess you have to try to input outside the physics before, because the physics affects also the particles.
I know. You're completely right, and it would be nice to work towards that goal. But what I'm saying here is that if my particles have a different, similar community when the flux is large, I don't care how it happened. You know?
No, but the effect of the community on the particle quality,
Yeah.
I guess.
Yeah.
And what I say, maybe you can put one of the positions here and put whatever they put to measure the vertical velocity. Maybe it's easy to.
Well, we will have the ACDP, but that's measure something. I have no idea. I know in Malaspina we were like crazy measuring, making sure the ACDP was measuring correctly and so on, and then I don't think I've seen many papers using this data for anything. So, but that thing that you say is very interesting, and students should look at the discussion that happened in LNO in, I don't know, 1979, 1980, in which Robert Peters and somebody else, I don't remember the name, made a big discussion about the role of empiricism and the role of experimentalism in ecology. And they felt like there were two papers, and then the responses, and there was a big fight among them. And Robert Peters said, I don't care what happens as long as the end result, I can predict the end result, I don't care about what's going on towards the end result. And the other one said, no, you have to understand, and you have to understand the mechanism, and blah, 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 blah. It's a very nice discussion, and it's very, I think it's very appropriate for students to see how some people working on the same feel about the ways of approaching. It's like the watchmaker piece-like thing versus the one that says, it gives me the time, so I don't care. As long as it gives me the time, I don't care what's going on inside. Well, if we predict it with this method, it will be a fast method to do the prediction. Yeah.
Okay, I think we can leave it here. I mean, we can stay here until tomorrow talking and discussing, but thank you very much for this very interesting talk and everybody for the nice debate.